Dear friends, here we are going to define one more important topic from phytohormones, namely mechanism of auxin action in plants. In this very topic, we are going to define the genes responsible for auxin synthesis, how they get expressed, how they stay on and off during expression. This is also called auxin signaling. Let me tell you terminology of the topic. Some important terms used in the topic are: number one, ligand. What is ligand? It is a basically signaling molecule, mainly in the form of auxin, used during the mechanism. Now, what is TIR1? It is a type of protein, or we can say it acts as a receptor for auxin. It is also called transport inhibition response 1 protein. This protein is a type of F-box protein family. It performs two important functions during this process. Number one, it acts as a receptor. It binds with the auxin. And second function is it acts as an enzyme, namely E3 ubiquitin ligase. Now coming to the third important point used in the terminology is AUX, or we can say IAA protein. It is a significant protein used in the mechanism. It acts as an inhibitor protein. And this protein stops expression of auxin genes, or we can say auxin proteins. This protein has two important primary functions. Number one, it stops transcription. It acts as a transcription repressor. Second function is it stops auxin gene expression. Now let me tell you how. Number fourth point to understand here is ARF, means auxin responsive factor. Now what happens? It is basically a transcription factor which leads to transcription of auxin genes, formation of auxin. Now AUX binds with ARF and it stops the expression of auxin genes, or stops whole process. Now to understand the mechanism beautifully, let me tell you symbolic representation of different proteins during the mechanism. Suppose, for example, for auxin ligand we can use symbol like this. Accordingly, for TIR1 protein we can use symbol like this. There are few more proteins AUX. Accordingly, ARF we can use figures like this. Later on we can apply these figures during the mechanism to understand it beautifully. For TRI1 enzyme we can use modified star like this. Now to understand the mechanism, let me tell you here, the mechanism of the whole process involves two pathways. Let us define case number one. What happens here? When auxin is totally absent, then AUX inhibitor gene, it binds with ARF, means transcriptional factor, and stops its activity. Auxin genes will not get expressed, whole signal stay off. Suppose this is DNA. These are auxin synthesizing genes present on it, and these genes, they get expressed, they get transcribed in presence of ARF factor, transcriptional factor. They form proteins. Later on proteins form what we called it as auxins. This is normal pathway, but what happened in case 1? ARF binds with inhibitor protein AUX in absence of auxin and form complex. It blocks whole pathway and keeps the gene expression totally off. This is case number 1. Coming to the case number 2, very important and very easy to understand here. Now it is reverse of first. When auxin is present in the process, what happens here? TRI acts as AUX, they both fuse in presence of auxin. Here auxin acts as a glue. Then ubiquitin ligase, which is actually TRI1 gene, it forms many ubiquinons called ubiquitin, polyubiquitin. Then many ubiquinones combine with these two proteins and form what we called it as proteasome. It is basically 26S type of proteasome. It breaks AUX protein, means inhibitor protein, which stops transcription. Then what is the result? ARF transcription factor is now free. It is dancing. It is ready to perform transcription normally. Let me show you here diagrammatic representation. This is DNA. These are auxin synthesizing genes. And this is transcriptional factor ARF. Now here it is a glue, basically auxin. It fuses TRI with AUX and form in presence of enzyme E ligase, then give rise what we call it as proteasome. Proteasome breaks AUX, that is the primary function. When AUX gets break, ARF is free. It expresses proteins, auxin genes get expressed, they form proteins, then proteins later on form auxins, and auxins perform different functions in the plant body. And here the whole process is on. It is the reverse of first. Hope you got the concept. Thank you. Have a nice day.